This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 242, recorded on July 18th, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today right here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
It's been a long time since you have been on TWiV.
I've missed it. I truly feel badly for not being on earlier and sooner and more frequently.
All right, and now that we've gotten past the prevarication, how do you really feel?
Miserable. Actually, it's hot as hell outside. It's really hot.
Yeah, good to have you back, Dickson.
Thank you.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How are you?
Doing okay. It is also blazing hot here.
What do you have?
We have 90 Fahrenheit, which is warm enough. Let's see, 32°C. You could almost use it as a warm room.
Yeah, we have 34°C here, which is the temperature at which we grow rhinoviruses.
Okay.
In the incubator, and that is that's probably mid-90s, I would guess, right? And
yeah.
It's also 54% humidity. Wow. Dickson,
Yeah, and it's only 11 o'clock in the morning because we're doing an atypical time.
We are.
Atypical. Yeah, supposedly it's going up to 36 here. So it's not going to reach 98.6, which would be 37. There you go. Also joining us today from the cool north central Florida, Rich Condit.
Howdy. How are you?
It is cool there, right?
It is cool here. It is 79 degrees Fahrenheit, which is a balmy 26 degrees centigrade.
Wow.
So I got you all beat. You know, it's good down here. I'm just trying to look up the dew point.
What is it going to be there today at the high point? Do you know?
The forecast is for 86 degrees with a 40% chance of thunderstorms. So actually, for this time of year, that's, a little cool. Right. For Florida, we could we could anticipate this time of year, between 90 and 93 degrees, but that's a little cool.
It is rather humid there, right?
Yeah, I can't. I'm blanking on the humidity right now, but it's a little humid, not bad.
Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Hey there.
Hey, Kathy.
What do you have there in terms of weather?
We have 87 degrees, which says it feels like 97 degrees. And
Right.
in Celsius, that's 29 Celsius.
Yeah.
So
Is it humid out there?
69% humidity. And so that's pretty humid. That's what raises it up to feeling like 97 degrees.
Wow.
And it's because it's art fairs in Ann Arbor. For those of you who have ever been in Ann Arbor in the summer, you know about art fairs.
So Kathy, what's the dew point? I got all my data here.
In all my 3 apps, I'm not getting the dew point.
Oh,
so,
bummer.
Yeah.
So it says it's now 81% humidity here, going down as the temperature rises to 63 when it gets up to 86 degrees. The current dew point is 73. Sounds like yours is similar.
Huh.
Yeah.
Now you're actually just southeast of hell, aren't you, Kathy?
Southeast. Yeah.
Okay.
It's west of us, right?
Is that a city in
it's
Michigan?
probably a little hotter than hell there.
Right.
Wow.
Right.
It's cool that you knew that, Alan.
Yeah.
Well, we are recording on a Thursday because tomorrow we will be traveling to, what's the city where Penn State is again?
Belfont.
State College.
State College.
Well,
Why would they name a place State College?
Well, there's a state college there.
It's the dominant feature of the town.
Is it the only feature?
I don't think it's the only feature.
And actually, the formal name is also University Park. So part of it. So
Well, I went to school in Ithaca, New York, where Cornell and Ithaca College are. They didn't call it College Park.
They just
but
have no
the University of
imagination.
University of Maryland is in College Park.
Is that
That's
right?
true. That
Yeah,
is true.
College Park, Maryland.
And
And there's
Texas
a branch of
A&M
the
is
University of Texas that's in College Station,
that's Texas
right?
A&M.
Yep.
That's Texas
All
A&M.
right, I
Texas.
stand chastised. However, I still can't remember where we're going. Hey, we're going there tomorrow to
Right.
the American Society for Virology annual meeting, which is a big shindig.
We meaning you and Rich, right?
Rich and
And
Kathy.
me.
Kathy.
Yeah.
Yeah, right. But I'm not going till Saturday.
So I'm going
And
Saturday.
I'm not going at all.
Neither am I.
Oh,
Actually, you could have. It's not too far for you. It's
No, actually, it wouldn't be too bad. But
oh well. I have to go tomorrow, so I have a meeting tomorrow evening. But we will be doing a TWiV there on Tuesday. It will not be live streamed, Kathy,
Okay.
and because the ASV tech is not up to the ASM tech. Maybe someday we will, but it will be released the following Sunday. And if you're going to ASV, you're probably not going to listen to this podcast anyway because you'll be going to meetings. So for the rest of the world, that's what we're doing.
ASV should just be absorbed into ASM, don't you think? Sort of endocytosed.
Oh,
no,
no.
there's a big topic for discussion.
Yeah, there was an exosome event years ago, and I don't
Yes.
think you're going to get it to.
No, I didn't mean to touch on it politically. That was
I think that it's unfortunate that there's very little virology at ASM. But they left years ago, the virologists, because they felt ASM was too big and they didn't get much attention. Now, this is an outstanding virology meeting. It's all virology for 4 days.
Yeah.
And it's really, really good. I really like it. There are ways that ASV and ASM could work together.
Well,
But,
ASP could throw themselves in there too then.
What's that? Parasitology?
Yeah, exactly, right. Just it's infectious microbes. Just do it.
But
well. If any of them ever need somebody to cover their meetings, they.
you never got an answer back, did you?
No, I've never heard from anybody there.
I'll follow up because you sometimes do have to follow up several times.
Yeah.
And speaking of follow-ups,
Aha.
Yes.
we have two. The first is from Carolyn Coyne, the author or the PI on the paper we did last week on human placental trophoblasts. And she writes, "Thank you so much for discussing our placental microRNA paper on TWiV. I am a bit delayed in listening. It is my listening material for my long runs each week, but I thought I would follow up on a few things in case you are interested." She has long runs in quotes, so I don't know what that means.
Hopefully, she means jogging, not norovirus.
I don't know if it's a 1 mile
You had to
or
say it, didn't you?
a 5
Yes.
or a 10 mile or a 20 mile. I don't know how long that would be. So an hour and 40 minutes, probably, if she gets through the whole TWiV. Juan, you were correct in that we did have some reasons for looking into the possible role of exosomes in the antiviral effect. This was very much due to the interests of my collaborator, Yoel Sadovsky, who had been looking into the role of exosomes and the C19MC-associated microRNAs in general placental development and during various pregnancy complications such as hypoxia. After we had excluded the obvious factors, which I initially was holding out hope for, such as type 1 interferons, we were scratching our heads a bit. Yoel was the first to suggest exosomes and microRNAs, and I distinctly remember that I was hesitant to think they were involved. Turns out he was right.
And much more interesting than interferons, I think. You
Yeah,
know,
I
it's
agree.
because it's new.
Yeah.
It's new. That's why it's good to have a fresh perspective, right?
Yep.
2, a similar hunch led us to autophagy. Again, after we had excluded interferons and some other pathways, we began looking at others. The student working on the project, Elizabeth Delorme Axford, was in the process of writing her doctoral comprehensive exam, which in her program is strictly off dissertation topic, and she was specifically focusing on autophagy and viruses. She suggested we add autophagy to our panel of possible pathways, and it was clear from the beginning that it was in fact playing a very prominent role.
So that's
There's an
now on
interesting-
dissertation topic, isn't it?
And
Indeed.
good,
Interesting bit of serendipity there.
a good reason to keep comprehensive exams in these programs. 3, we are following up on the targets of the microRNAs and have recently started collaborating on work with CMV. So far, we don't have many answers, but it is still early days. And 4, I absolutely agree, RE, the figures. It is tough packing so much into the strict page limits. I felt the overall paper suffered from it. Thank you again for your interest and support, and I look forward to seeing you at ASV. A student now working on the project is presenting some of the follow-up work. screening some additional viruses as well as Toxoplasma gondii.
Okay.
In a talk on one of the first sessions, Workshop 3, if you are interested.
Nice.
Toxoplasma.
Well, it harbors double-stranded RNA viruses.
It does, Dickson. We did one of those on TWiV, right?
We did that.
I think this is cool how the PI can come to us and talk about these kinds of off-target things. Isn't that cool? It's great.
I think that's one of the real bits of magic about this whole thing is getting the PI's input on the behind-the-scenes thing.
That's right.
I don't think you could do this any other way, and it gives great in-depth understanding and ideas for the next set of experiments as well.
I think this allows the public to see how science is done.
That's right.
And this can only be done on a long format show like this, where you have plenty of time, and there's not a producer over there waving cut.
Your sound bites are too long.
That's
Really?
right. That's right. Could you repeat that now in fewer words? No.
All right, Robin writes about placental exosomes. Are exosomes sufficiently antigenic to be promptly cleared from the maternal circulation? Would they confer viral resistance to the mother? Would they be involved in maternal immune tolerance to the fetus?
Don't know.
Duh.
All interesting questions.
Yeah.
Well, the exosomes are produced from the placental.
Right, so they are
trophoblast.
baby.
Right.
Yeah, so I don't know. How does that work? Is it recognized? There is some recognition as baby, right? That's actually why. Well, that's earlier on and during implantation. It's actually good to have an immune response.
I think we would have to ask Carolyn's collaborator
Yeah.
some of these questions. So,
Yeah.
so you all come back now.
Viral
Oh,
resistance.
bad pun.
Really?
Y'all.
You know,
Oh, is that his name?
yeah.
Yes.
See, I've already forgotten. I can't remember names.
God, I missed this show.
Would they confer viral resistance to the mother? I would. Why not? Right?
Assuming that those exosomes can actually target maternal cells.
Mm-hmm.
All right, and I don't know how exosomes.
Yeah, I gather
done.
the point of that system seems to be to keep a virus that's already in the mother from getting across the placenta, right?
Right. Or at least that's our anthropomorphic notion of what the point is.
So toxoplasma can cross the placenta despite the exosomes, and maybe that's why they're looking at them.
Yeah.
Cool stuff, but it's only a double-stranded RNA virus that it can transport. It's sort of like a paratenic host, but that wouldn't apply to other viruses, I don't think, would it?
Well,
Or would it?
any virus that gets across that's vertically transmitted across the placenta is going to have to get through this barrier in some way at some frequency, right?
So maybe
Right, and that's why CMV is interesting.
maybe
That's why CMV is interesting, yeah.
Maybe toxo drags it in with itself.
I don't know the answer to the tolerance. I'll have to send these along to Carolyn.
Yeah.
We can go back and forth. He says about MHC in devils. If absence of the MHC confers immunotolerance in the Tasmanian devil cancer, can this be useful in organ transplants?
Huh.
Absolutely. It is my understanding that for example, we put pig parts into people, right?
We do.
Right.
Heart valves.
That's right.
And they're working on making transgenic pigs that don't have MHC.
Chimeric.
And I know just enough immunology to be dangerous. So the first thing that I think about is, okay, if you have cells that have low or no MHC, then they're a nice target for natural killer cells. So maybe that's going to be a problem.
Could be.
I don't know.
I don't
And
know.
isn't this is a little bit of a digression, I think, but in the pigs, isn't there also a sugar that needs to be changed?
a sugar?
Yeah, there's a cell surface glycosylation that's I won't say the chemical names because I'm sure I'll get them wrong, but it's a particular sugar that's found on the pig cells that's not found on human cells.
Nope.
I seem to recall something about that.
Don't know. You're out of
But
my
yeah,
head.
the MHC is another.
It's cool. Yeah, it is.
Another target.
It is something that people are looking at. But of course, if you transplant from one person to another, there's not much you can do about MHC
Right.
Right.
unless you infect with a virus that knocks down MHC or something.
Well, you give them cyclosporin A and hope for the best.
Well, that's what they
Right.
do now, right?
Yeah.
Yeah.
Yeah, and in fact, in the original transplants, some of the original transplants, they gave the immunosuppressives initially and then tapered them off, and people became tolerized to the transplanted organs. But now the approach is to just pull out the big guns and dose people up with the immunosuppressives their whole lives, because you may or may not develop tolerance.
Yeah.
All right. Today's paper is actually... So last year at ASV, Carolyn Coyne gave a talk about the placental trophoblast study, which we did last week, and she was on TWiV. And Sarah Sawyer also gave a talk about virus receptor arms races, and she was on TWiV, and her paper has come out recently, so I thought we would do her paper. It's kind of an ASV week.
Give everybody their TWiV bumps before the conference.
Yeah. So her paper came out in PLOS Biology in May this year, and it's called Dual Host Virus Arms Races Shape an Essential Housekeeping Protein by Dimogenes, Abraham, Choe, Farzan, and Sawyer. This is Sawyer's lab, of course, at the University of Texas, Austin, which Rich and I will be visiting in the fall.
Good.
In October.
You remember that, Rich, right?
I got it on my calendar.
Yeah, that should be interesting. I've never been to Austin. I hear it's a good place.
That's a wonderful
Nice
city.
place.
Wonderful city.
So this is a very cool story about a cell surface protein, the transferrin receptor, which of course is important for iron, regulating iron uptake, right, Dickson?
Yes.
And it happens to be also the receptor for 3 different families of viruses. And this includes arenaviruses, retroviruses, mouse mammary tumor virus, and parvoviruses. And this paper looks at how there's a conflict between viruses and receptors. So we've talked a lot about this virus-host conflict and how it's been studied by the Malik and the Emmerman labs. We've had podcasts with all of them. And and most of those studies have to do with defense proteins overcoming virus infections and the viruses mutating, to overcome them in turn. This is a virus-receptor interaction, which you can imagine can undergo the same back and forth, right?
You bet.
And so that's what they're looking at here. So they're saying, can we detect a conflict between, in this case, a receptor which is utilized or bound by several different viruses? All right, so the way you do this is very interesting and actually is explained really well in this paper. Most genes undergo change, mutagenic change, a lot in ways that don't change the protein sequence. Right? And these are called synonymous mutations. But they also undergo change.
Just to elaborate on that, the genetic code is degenerate, which means that there are multiple codons. I mean, there's 64 possible codons. 3 of them are stop codons, so that's 61 coding triplet codons,
for 21
and there's only 20 amino acids. So there's on the order, on average, of a threefold redundancy, which means you can have on the order of 3 codons to encode a single amino acid. And typically, it's the third position, and there's a molecular mechanism for this, the third position in the codon that you can change without changing the coding of that codon for an amino acid. So in evolution, that nucleotide is free to drift because it doesn't change the protein composition. So that can be regarded as sort of, a measure of what the random mutation rate is that's not under any kind of selection.
Right.
Non-synonymous. I'm sorry, synonymous.
Synonymous, yes.
Right.
So, and just to do it sort of as this week in vocabulary, it comes from the word synonym, where you have 2 words that mean exactly the same thing.
Ah, that's
So
great.
you have 2 codons that code
didn't know
exactly the same amino acid.
Thank you. I was going to ask, where does the name come from?
There you have it.
Who did that? Years ago, or was it a recent thing? Do you know, Kathy?
I think it's years ago.
Yeah?
Yeah.
Because the bioinformaticians have taken it over.
Yeah.
Yeah, I think I remember learning that back before there was such a thing as bioinformatics.
Now, on the other hand, the non-synonymous mutations are those that cause amino acid changes, and those a lot of those are going to be deleterious to the protein. It's going
Right.
to mess up the protein function. So you don't see those to the same extent as the synonymous mutations. And the idea is if you programs have now been written that can scan sequences of genes and identify residues that have more variation, have a higher non-synonymous to synonymous ratio. And you can do this not only in genes from a single organism or species, but in multiple. And in this case, they looked in 7 different rodent species, which have a transferrin receptor, of course, and they also harbor various viruses. And you can say, are there certain residues that have a high mutation frequency? And those are called they're said to be under positive selection because there was some pressure put on at some point in evolution that caused a change in the amino acid sequence.
And they're actually drifting at a rate that is in excess of the synonymous mutations. So it really does look like positive selection. They're changing for a reason, it appears.
Yeah, so here in this transferrin receptor, they found codons that have a non-synonymous to synonymous rate of mutation of 4.2. So neutral evolution would be 1 and less than 1, but greater than 1 is, you look at that as being positively selected.
Well, I think right. Drift. You'd have non-synonymous over synonymous would be a lot less than 1.
Yeah. Mm-hmm. So anything like greater than 1, I think. I don't know what the cutoff is, but in this paper,
if it's greater, they said greater than 1.
Yeah.
Is their cutoff for positive selection. And in this particular thing, what they're seeing is positive selection that's in specific places within this gene.
Right.
Mm-hmm. Yep. So for me, the interesting thing about this whole deal is that here's a set of viruses that makes use of a functional protein that we can't live without.
Right.
Yeah.
So that other receptors are not like that.
Not all. So some are immune. Well, some
are sugar
virus
residues
receptors. I think most of those are quite. CCR5 comes to mind as non-essential, right? You can delete that part of that gene in people, and they're okay. And offhand, I can't think of others. But the immune defense proteins,
right?
Some of those can be taken away, and the only problem you have apparently is you can't defend against the virus.
Right.
But receptors, immunoglobulin-like receptors, I think most of those are pretty important.
Okay.
Anyway, you okay with this so far, Dickson?
I'm very okay with this.
So the cool thing here is, of course, they have the crystal structure of the transferrin receptor, and they also they happen to know the parts of it that interact with arenaviruses and MMTV.
Yeah, this is a nice system because people have studied the heck out of this receptor.
So they identify 6 codons in the receptor that are highly positively selected by virtue of this non-synonymous to synonymous ratio of greater than 1. And these 6 residues are on an external part of the protein. 3 of them are, and they map to the known virus binding sites for these 2 viruses, MMTV and arenaviruses. And they have a beautiful picture that shows that. And the 2 virus binding sites are separate on one side of the transferrin receptor. I'm going to show this to you, Dickson.
I'm looking at it. It's beautiful. But they're not. They don't take advantage of the active portion of the molecule that the host needs in order to survive.
Right. So these changes obviously preserve transferrin receptor activity because they're there and the animals are fine and so forth. So again, 7 different rodent species. Here you go, Dickson.
Got it.
See, arenavirus binding site,
Yeah.
it's separate from the MMTV binding. And so that's so cool that these are the sites identified by this positive selection algorithm.
So, Alan, I think the name of this podcast should be I Want My MMTV.
Ooh.
That's good.
Not bad at all.
That's a good one.
Not bad at all. By the way, these 3 different viruses that use this transferrin receptor are completely different viruses. MMTV is a retrovirus. The arenaviruses are basically negative sense segmented genomes. Actually, they're ambisense to be a little more specific. And the parvoviruses are single-stranded DNA viruses. The capsids are different. They couldn't be more different. and they're all using the same receptor, though different parts of the receptor.
Right. And the rodents include the house mouse, the brown rat, which I
Right.
know you're very fond of, Dickson.
Absolutely.
The striped dwarf hamster,
Ooh.
the short-tailed zygodont, the common
Which sounds like a dinosaur.
It does.
I love it. Yeah.
the common neocomes, the vesper mouse, which I love. Does anyone know what a vesper is?
Well, it's something to do with church.
You guys think
It's in
James
the-
Bond. Think James Bond.
Vesper?
Yeah.
Oh, is it a car or something, or no?
It's the name.
Bad
it's
guy.
the name
There-
of one of his martinis. It's called a vesper.
Yeah, but it's a religious thing, though. I'm sure.
Yeah, Vespers is a
Yeah, it's
a prayer.
That's right.
And the drylands laucha.
You see, we should keep some of these names handy to call other people when we really want to insult them without them knowing that they're being insulted.
Speaking of which, what's Vincent got? Well, I mean, sorry, what's Dickson? What's Dickson's association with the brown rat?
Natalensis?
He got bitten by one in New Orleans.
He did.
when he
Is that
was-
right?
Oh, yeah, I can show you the scar from it.
No, we don't need to see this.
You don't need to see the scar?
on your
Okay.
finger, right?
Yeah, it's on my finger.
He told this story on a TWiP a long time ago.
I did.
And
I
it
did.
shaped his being. This is why he is the way he is today.
Is that right?
Yeah.
No, that's not
Okay.
true at all.
The
Come on.
brown rat, Rattus norvegicus, right?
That's right. Rattus norvegicus.
So these are the 7 that they sequenced. They analyzed the sequences of the transferrin receptor and used their algorithm to find these positively selected residues. So some of these are susceptible
Right.
to these viruses and some are not.
Yes.
So this is really cool.
So, Alan, another thing just for Alan here.
Yeah.
If the virus were to have attached at the active site of the molecule, then we would all be in dire straits.
If the virus were smart, it would bind to the active site, because then the host can't change that without
Correct.
sacrificing the function of that receptor.
Yeah, well, exactly. Well, I meant that as a reference to I want my MTV.
My MMTV,
My
yes.
MMTV.
Now you can do the same kind of analysis with viral gene sequences, this DNDS type analysis.
yeah.
And they do that with in the case of the arenavirus, the GP, the glycoprotein that attaches to the receptor. So you might expect to find positively selected residues there, right?
Exactly, sure.
And they do. They find
Wow.
residues in the glycoprotein that interact with the transferrin receptor that are under positive selection. I think 11 codons have DNDS greater than 1, and 10 of these correspond to surface-exposed residues on the glycoprotein structure.
Wow.
So this is just beautiful, right?
Yep.
Now, Rich, didn't Mike Emmerman say that sometimes you can't do this because the viruses change so much?
Yes, I believe that's the
And
case.
in fact, that's why they look at host genes, because the viruses change so much that you can't often
right.
pick up these signatures. But here in this case,
In this
it
case,
works.
apparently can. And I gather from this that the arenavirus receptor interaction and the different species of arenaviruses interacting with a receptor is stuff that has been done over time before this paper, and that's pretty well studied. That
Yeah.
there's specificity between these different arenaviruses and the different receptor sequences that
Right.
makes sense in terms of this variation.
Right. Now, I don't know if I should go into this. Turns out that this only one of these 7 rodents is known to be infected by MMTV, so they were a little puzzled as to why there was positive selection on the MMTV binding site of this receptor. So they looked in the genome of some of these mice, and they found endogenous MMTVs, which suggested that these viruses have circulated at some point earlier in some of these species, which might have explained this selection.
Right. So it's as if the host is actually winning here.
Yeah.
To the point where the changes have restricted the MMTV to simply one species of rodent, whereas previously there were other species that were bothered by MMTV.
Yeah, well,
And-
the other way to look at it, though, is that all the rodents had to have a common ancestor sometime in the distant past, so maybe it started out as one, and then as the rodents radiated species, it was retained.
Dixon, what is the oldest rodent? Do we know?
We don't.
Species?
We don't.
No?
No.
Okay.
But apparently, some of the early mammals, back maybe 60 million years ago, just after the big bang of that meteor that hit the Earth, so those were rodent-like mammals that survived because they burrowed into the ground. and survived all of that Holocaust.
Wow.
So maybe the earliest rodents date right back to the age of the dinosaurs. Because there were mammals running around when dinosaurs were still present.
They're running around their feet, right?
Around their feet, picking up scraps.
Now, one of the things now you can do is to take these parts of the transferrin receptor and swap them between susceptible and resistant hosts. So they do that for both MMTV and the arena virus.
Cool.
So for the MMTV, they take these residues under positive selection, the three positions, and they swap them from a susceptible to a resistant mouse, and then they express the proteins in cell culture, and they show that.
Wow.
You put the right mutation in, you can get infection. If you put the resistance in, you get the resistant amino acid in, you don't get infection.
It's like a molecular Koch's postulates.
These are really elegant experiments.
That's fantastic.
And it's so neat because
good stuff.
Just the mutations at 3 residues are enough to regulate the virus entry, but they keep the function of the transferrin receptor.
Right. That they do look at that, actually. They look at transferrin binding, and they show that none of these changes affect it, which
it's amazing.
Makes sense because they're in the genomes of these animals that are around.
And as you would predict from everything else that's gone on, it affects the binding of one virus but not the others. So MMTV and not the arena viruses.
Right. They did this also for the arena. Is that right? I'm not finding.
They did it for one of the arenas, I think.
Yeah. Okay. Now, Dickson, do you think that the human transferrin receptor will at some point be in an arms race with arena viruses?
If we are susceptible to arena viruses, the answer is yes.
They are beginning to new world arenas are emerging into people.
Sure, why not?
And this is mainly in South America, where as we impinge upon the forest, we get infected.
What's the consequence of infection with arena virus?
Well, you can get these hemorrhagic fevers.
Really?
Not good. Loss of fever.
Okay.
Argentine hemorrhagic fever.
Of course.
These are bad things.
Yeah, don't get this.
Case fatality. So Hunin virus, which causes Argentine hemorrhagic fever, has a 20% case fatality rate, Dickson.
Right. That's pretty severe.
So if people have polymorphisms, amino acid changes in the transferrin receptor that make them a little more resistant, they would survive, and eventually that would, right? It'd take thousands of years for those.
I think you'd need the infections to be common enough.
That's right.
That they would have a real impact on people's reproductive rates.
Well, with Lassa fever, you could probably do that survey right now because it's a very common infection. Apparently, they have annual outbreaks in West Africa, so you could look.
So the problem here is that, of course, with human interventions, we can allow people to survive that don't have these polymorphisms.
And we've been in Africa for several million years.
I don't know what's going to... Anyway, in theory, these mutations would be selected for. So they went and looked in the human genome sequences to see if they can find any polymorphisms in the transferrin receptor. And they found one at position 212, which is close to their positive selected residues. It's a leucine to valine change. And they put it into transferrin receptor, and they say that it confers some resistance to infection. It's not completely blocked, but there's some resistance.
What about non-human primates?
Non-human primates.
In other words, looking for their sequences.
Yeah, exactly right.
They
don't report on that.
I don't recall.
Okay.
It's a good question.
I would say, West African non-human primates might be worth looking into.
This polymorphism, by the way, is found in, I think, Asian human sequences. And there are no known New World arenas infecting them.
Interesting.
So what do you think about that, Dickson?
Well, it indicates that we migrated from some place that used to have them.
Or that it's just a random mutation, right, that happens to be near the virus binding site.
Could.
So if arenas ever made it into Asia, maybe this would be selected for.
Right.
But at the moment, I mean, there can be neutral amino acid changes, right?
Right. And by the way, Lassa is a rat parasite or a rat virus.
Mastomus natalensis.
That's the one, the multimammate rat.
Multimammate.
That's correct.
Rats are normally not multimammate?
No, it's just they all are, but this one is called that because it's obviously multimammate.
So that's the story. It's very cool.
Very, very nice.
Yeah.
Love it.
They say it's the first case, to our knowledge, where the evolution of a single host gene is driven by 2 host virus arms races at once.
Wow.
So now you could take the same approach and look at, viruses need a lot of different cell proteins, right?
Sure.
So this is what I was thinking. You could look at lots of them for
Right.
signs of positive selection.
Right, sure.
And do you think there will be some where there are none because you cannot, the function that the virus and the host utilize overlap so much that you cannot do this, like a ribosomal protein maybe?
Sure.
Right.
Right.
Yeah.
Right.
And then there will be others like, I mean, actually, I guess we shouldn't be surprised that a transferrin receptor could have residues that aren't involved in its function that could be
Right.
binding. And viruses have selected that, right?
It also raises the issue of species extinction and how that occurs, and could that be driven by certain introduced viruses into an area where this receptor is totally susceptible in virtually every individual?
It's interesting to me that the viruses, bind to these sequences that are subject to some variation. It seems like a stupid thing to do. there must be a reason for that.
Well, the virus is constrained and probably can't modify the I mean, it's not just the transferrin receptor that's constrained without killing the host. It's the virus that can only assume certain shapes, and so it capitalized on this receptor, and maybe it can't bind to the active site of transferrin receptor.
Well, what's the steric picture look like with regards to
If you look at Figure 6, they've got a nice figure of the variable sequences, the drifting sequences in red, and it's bound to the transferrin receptor in purple. It's completely non-overlapping.
Right. But, I mean, when it's inserted into the membrane, how does that actually look in terms of what the virus encounters first?
Well, I think the bottom here is where it's attached to the membrane.
That's the...
But I don't really see why the virus could not evolve to. I guess there is some reason that we don't understand, but...
Yeah, otherwise it would have won.
Right, but so it obviously evolved to this portion for structural reasons, constraints, as Alan said.
Sure.
And it just happens to be apart from the regions that are important for transferrin binding. But it could go either way, right?
Seems like that to me.
Yeah.
I mean, I think that you would favor binding to sites that can be changed so that the virus could. Well, no, that doesn't make any sense.
No, because that works against you.
Yeah, it works against the virus, but for the host, right?
If you were to
these may have initially just been the most accessible sites
That's what I said.
for the virus, and then once it went down that evolutionary path, it couldn't get back.
Yeah.
If you were to insert a picture of the real virus next to that molecule, it would be quite large compared to the molecule. So, it's like watching the Hindenburg land in Lakehurst. The pinnacle of the landing spot is that receptor site, but the blimp itself couldn't possibly fit inside that active site. So maybe this gigantic particle virus just can't have access to those other parts. The interesting thing here is that for viruses that bind to immunoglobulin-like proteins, like poliovirus and reovirus, the parts that they bind to are analogous to the variable regions in antibody molecules. So there, it's closer to the active site. And so maybe this kind of study with those sorts of receptors would illuminate some cases where the functions overlap more. I'm sure there will be some. It's a Lego solution.
It's just early days, as she says.
Early days, right? Early days.
I wanted to introduce one last concept here, which I found good, and everyone should know this, and that is a hypomorphic mutation.
Hypomorphic.
So we've talked about mutations in the receptor, the structural protein that affect virus binding. But there can be mutations in other genes that control the expression of, say, the receptor protein and lower its sufficiency so the virus can't infect. So there's no alteration structurally in the receptor, but its expression level is changed. And those are hypomorphic mutations. So I thought everyone would like to know that.
That's a
good term.
Yeah,
hypomorphic.
Yeah. Like it.
It's a genetic term. The geneticists are great at making terms.
Yes. And if you upregulated the receptor for some purpose, would that be a hypermorphic mutation?
So.
Okay.
It would be.
Yeah. Hypo meaning under, like a hypodermic syringe.
Right.
Easy way to remember the...
Less than.
Oh, I never thought of it that way.
I never thought of it that way.
Under the skin.
You should come to my virology class. I'm constantly giving them ways to remember these vocabulary words.
Well, it's too hot and there are no weekends in Ann Arbor, but
Right.
I'll listen to you. All right, so that's that paper. I thought that was cool. Thank you, Sarah
Very
and
cool.
company.
Yeah.
And, what's this picture of a mouse? A zygodont?
Yes. And it's from the ASM webpage, meaning American Society of Mammalogists.
Yeah, they have
Oh,
the same.
he's a cute little critter.
Yep.
Yeah.
Wow,
That's right.
zygodont. All right, thank you. Oh, by the way, so I wrote a blog post about this yesterday, and there was one comment already, and this is a great comment, which I thought we could address. Why don't long-lived species like, say, redwood trees succumb to viruses? Surely they have no chance to mutate quickly enough to participate in the arms race.
Well, I made a casual observation that most parts of a tree are dead anyway, so it wouldn't be susceptible to a virus infection to begin with. But maybe there are some natural chemical compounds that just are totally protective with regards to any kind of invasive organism like sap.
The problem is that if the tree has survived, it's got the right mutation to survive, right?
Yeah.
So he's saying if the virus changes,
I
why doesn't it
understand.
kill the tree?
Understood.
You know, my impression is that many of these plant viruses are good.
Tell him the Dutch elm disease was a fungal infection, but nonetheless, it did
There are
knock off a whole bunch of
there are
trees.
many tree viruses.
Yes.
Yeah.
And I think that the reason they haven't wiped out the trees is that they are on the same evolutionary schedule as their hosts.
Aha.
So it's not, we think of redwood trees as slow growing and viruses as fast growing, but that's because we're thinking of viruses growing in HeLa cells overnight or in bacteria that multiply every 20 minutes. But a virus that grows in a redwood tree is going to be limited in its spread to the speed of its host's metabolism.
Yeah.
So if the host metabolism is slow, I would think the virus metabolism would be slow.
Right. Well, I would have picked the bristlecone pine tree anyway as an example of a long
Sure,
lived
that's a very long-lived. Sequoias and redwoods get huge, but
Yeah.
something like a bristlecone pine lives for centuries or
That has-
millennia.
It has a very narrow strip of living tissue and a lot of dead wood. So
Yes, there's a lot of dead wood that's not going to be hurt by anything.
That's true.
But the living tissue is not growing very
Yeah,
fast.
that's also true.
So I think that may be part of it. And there are antiviral defenses in plants.
Right.
They don't have antibodies and the sorts of immune systems that we have, but they have certain defenses against viruses, so some evolution has occurred there.
Sure, and insects too. They even produce insecticides and insect
Sure.
repellents and all kinds of other things.
But he or she says, why don't long-lived species succumb to virus? How do we know they
Yeah,
don't?
well, that's right. Whales, for instance, a whale.
Well, there are viral
elephants.
diseases of trees that will kill trees
Yeah.
under the right circumstances, so
of
they
course.
can succumb to viruses. Yes.
Right.
And the ones that are living just haven't, right? But
Right, they haven't been driven extinct because of the usual dynamic of virus host
That's right. That's right.
pressures.
all right, let's do some email. Dickson, if you log into your Google Doc account on that computer, which is right next to you, you could see the emails too. Do you know how to do that?
You're going to tell me, aren't you?
Well, first you have to launch a browser. But in the meantime, Alan, could you start with the first email?
Okay, and I'll just warn Dickson, when you scroll down, don't be scared when a giant zygodont jumps
Oh, thank you.
All right, Aaron writes, just finished listening to episode 227, where several vaccine disinformation topics were discussed in the emails. Sincere thanks for your great show and all your great work. Here's my question. The tobacco cancer disinformation campaign was covertly funded by the tobacco industry. The global warming disinformation campaign is covertly funded by the fossil fuel industry. However, when you look at the anti-evolution disinformation campaign, it seems to just be driven by a bunch of zealots. Regarding the anti-vaccination campaign, is there some fraudulent/holistic healthcare industry money behind it, or are we simply dealing with zealots? I feel like it would help to have this information because pointing out the behind-the-scenes profit motive can be a powerful resource when dealing with disinformation. If we're just dealing with earnest but misinformed people, there's a more daunting challenge since it means there is a fundamental distrust of the medical community. Sorry for the downer of a question, but I really don't know the answer to this one. Aaron is in Maryland. And I would point him to a book called Autism's False Prophets by Paul Offit. Offit does an excellent job of describing the cesspool of money and greed and just there's a whole industry built around selling snake oil,
Right.
essentially.
Right.
And the people in that industry are largely responsible for the vaccine disinformation because they want people to go and buy what they're peddling, whether it's lawsuits against vaccine companies or some sort of quack treatment for their autistic kids. And autism is just that's the topic of Offit's book because that's how it really came to the fore in the vaccine area, but that overlaps with an entire pit of this sort of thing that goes on in the so-called alternative health industry, which is a multi-billion-dollar industry. And the anti-vaccine thing, Andrew Wakefield's case is particularly illustrative. He was actually in the pay of lawyers in the UK who set up that study and funded it for the purpose of filing lawsuits against vaccine companies.
All right.
So yeah, follow the money. and so thanks to some incredible investigative journalism by people like Brian Deere, we now know the story behind parts of this. So read Autism's False Prophets, and you'll see where a lot of that comes from. A lot of the people are just zealots because they're parents with kids who have problems that they haven't been able to fix. But the real driver behind it is, yes, there's a lot of money on the table.
I think I'm going to be sick.
Yeah.
I haven't actually read this book. I've just started reading the book on vaccination on Hillerman. And so I'm really behind on my TWiV reading, and I must read this.
Autism's False Prophets. I had put off reading it because I thought, this is just going to make me mad, and it did. But it was morbidly fascinating.
Mm-hmm.
I mean, you're reading along, you're thinking, my God, these people, how do they sleep at night?
Yeah.
And it's really, really astonishing.
They sleep on a cushion of money.
On a very large, comfortable bed, I'm sure.
That's right.
Rich, this article you linked to, you have to mention this. This is ridiculous.
Yeah. So recently, Jenny McCarthy has been hired by ABC News as the anchor for their show called The View, which is I don't really know much about the show. It's a kind of a talk show that I guess looks at current events of one sort or another. And Jenny McCarthy, for those of the listeners who don't know, is first came to fame as a Playboy bunny, and she is described as very telegenic. She looks good on the TV and has made a name for herself as one of the real anti-vaccine crusaders. She has a son who has autism, though actually it's been questioned whether his problem is actually autism or not, but that's neither here nor there. And she's taken up the crusade about the anti-vaccination crusade, and unfortunately now she has this platform. We'll see what she does with it on ABC. And one thing I really like is that if you do a Google search recently for Jenny McCarthy, what you find out is that a number of different news outlets have bashed this decision by ABC News to give her this platform.
Mm-hmm. Really stupid move by ABC.
Really stupid move by ABC. Really stupid move. And it will be interesting to see what happens. Probably nothing, unfortunately, because a lot of people listen to this woman and take her seriously. And
well, she has a nice face. You know, that's what got her there, and that's given her a platform, and she has become the celebrity spokesperson for this anti-vaccine movement. I think she probably falls into the category of just a zealot. I don't think she's actually making money from her anti-vaccination stuff. She's making money from the fact that she's a celebrity.
Though her celebrity status has been significantly enhanced, I think, by the controversy over the...
Enhanced and damaged at the same time. It's hard to say whether this benefited.
Mm-hmm.
One thing that's interesting is on the first page of Google Hits for her is the site that's called Jenny McCarthy Body Count.
Yes.
Right.
Which is counting the number of vaccine-preventable deaths and illnesses since June 2007 when she started her anti-vaccination campaign.
Very good.
Yeah.
So...
Currently 118,000 preventable illnesses and 1,170 preventable deaths.
And
so if
no
I'll go to
deaths.
that link. It might move up on the Google Hits page. So,
All right. Well, there's no such thing as bad publicity, right, Alan?
That's right. Well,
Only if they spell your name wrong.
All right. Let me take the next one because it sort of has to do with TWiV. James writes, Kia ora et al. Now, what's that mean?
hi in New Zealand.
New Zealand. Oh,
Yeah, it's a Maori language greeting.
nice.
Really?
Kathy,
I didn't know that.
where did you get that?
I'm sure you Googled it, right?
Well, Kia
did.
is their bird.
Okay.
That's a
Very
parrot.
good.
That's a flightless parrot in New Zealand. So maybe that's ora is the way it speaks.
Kia. It's also a bad car.
Right.
yes.
And worse than yours, Dickson.
Ah, come on. Mine's not a car. It's a vehicle.
It's a fraud.
It's a Toyota Prius.
It's a fraud.
It's not a fraud.
In a recent TWiV, Vincent mentioned how he'd like to save up some more funds for new equipment. As these tend to be one-off capital expenditures, perhaps Vincent would like to try some crowdfunding for specific items. There are a bunch of sites out there now to do this sort of thing. Indiegogo seems to have become one of the big ones and seems like it would suit Vincent's need. Disclosure, I do not work for Indiegogo and have not donated anything to something using it, though for TWiV, I'd be more than happy to throw a bit of cash into the pot. P.S. I would say no need to read this on the show, but I know you will. Anyways, James is a software developer in New Zealand, and that's a great idea, and I will be doing this at some point.
Yeah.
So thank
Cool.
you, James.
Yeah.
Funny.
Kathy, would you take the next one, please?
Michael writes, to Vince, Kathy, Rich, Alan, et al. I just realized that my last email, great paper stem cells used to identify CNS cell types susceptible to HSV, was addressed to great men of TWiV. This was the salutation I used last time I wrote to the show a year earlier. However, it's no longer appropriate. If you read it on the show, please amend it to great humans of TWiV or the like. I also want to draw your attention to the latest editorial on the oversupply of trainees in the biomedical sciences, The Writing on the Wall by Henry Bourne of UCSF in eLife, and he gives a link to that. He says, in one of your recent interviews, I believe it was with Ian Lipkin, the interviewee said that he got his first postdoc job by writing letters to a bunch of people he admired all over the world, asking if he could join their lab. Something like 24 out of 28 responded by saying, sure, come and work here. And he had to be careful in choosing the right one. Nowadays, it would more likely be 1 out of 28.
Wow.
Best wishes, Michael.
Wow.
This is a good article. I like it
Yeah.
very much.
Yeah.
Has a lot of good ideas in it. I highly recommend it.
What's interesting to me, though, is when I was coming out of graduate school in the mid-'90s, there was a spate of articles virtually identical to this one, talking about how there was an oversupply of PhDs and the job market wouldn't be able to absorb them all, and we would need to do something about this. And there was a lot of fretting and hand-wringing. And then the NIH budget doubling campaign got underway, and everybody forgot about all that.
Mm-hmm.
As if doubling the NIH budget was going to cure all the problems.
Right.
Which, of course, it didn't.
And one of the things that he points out in this that's really very interesting is that one of the reasons that doubling the NIH budget doesn't really help is that the money, too much of the money gets spent on building infrastructure and that kind of stuff that can't be sustained once the doubling stops.
How about that?
So we've had flat or declining NIH budgets for years now. It's clear that this isn't going up anytime soon again.
Yeah.
Right.
We have to work with what we have. I mean, $30 billion, we have to work with that. It's not bad. And he gives a number of good suggestions, and it just shows that the universities have to help.
Yeah.
They have to pay salaries. The indirect costs have to be fixed and so forth. I mean, the university has gotten really a lot of money from NIH for many years. They really have to start kicking in.
The problem is that there's a whole set of dependencies that have been built on top of this over the past 50 years.
Yeah.
You bet.
That's right.
And universities, to Columbia, Vincent, you're a cash cow.
That's right.
They hire faculty members for the purpose in the sciences for the purpose of bringing in grant money.
You bet.
And if you make the university kick more of that money back to support their own faculty, then the cash cow goes away, and they've got to figure out some other way to run the rest of the university.
Right.
So it's a huge, huge, deeply entrenched problem.
Not going to change overnight, I understand.
No.
But you have to start now.
You do have to start, and
changes.
the NIH has the power to do this.
Right.
They can say, okay, we're only going to allow X amount in indirect costs.
Yeah, well,
And they can define X to whatever they want it to be.
but remember, Alan, the Office of Naval Research was the organization that was used to negotiate those overhead costs.
Yeah.
No kidding. So there are layers upon layers upon layers. I was a cash cow. I'm now out to pasture, but
Now you're a cow. Now you're just a cow.
that's right. I mean, they didn't care if I did get my grant. They cared if I didn't get my
Yeah.
grant, and that's the onerous part of being one of these cash cows.
Anyway, read the article. It's really, really good, I think.
Yeah.
Right.
Rich, can you take the next one?
Sure. Stephen writes, I just listened to part of TWiV 21 to get a little more on polydnaviruses. I know you mentioned polydnaviruses on one later episode, but it'd be great if you do a whole episode on them with an expert guest. By the way, most illustrations show brachnoid wasps laying eggs on large caterpillars, but the story is a little stranger. Some brachnoids lay on first instar larvae hidden in flowers. Here's an image I submitted to bugguide.net, and he has a link to that.
Right.
Bob Carlson comments, according to Sharkey 2006, most agathindinae are solitary attack first instar Lepidoptera larvae in concealed microhabitats such as leaf rolls or stems and emerge from the last larval instar after it has spun its cocoon.
Ooh.
And
Get
he
them
gives
when
a
they're
link to us.
sleeping.
Oh, my goodness. You have to wonder how widespread polydnaviruses are.
You must wonder.
So we actually did a polydnavirus paper on, actually a paper and some associated papers on TWiV 179, and we had a pretty thorough discussion there. It still wouldn't hurt to get a polydnavirus person on the show sometime. They're fascinating viruses, but that's another episode you can check out to see it. And I like his pictures here.
Is it Michael Strand who was at ASV last year?
Yes, Mike Strand. He gave an ASV talk last year. We actually had him here for a seminar, and it was his paper that we did on the show that was some sequence analysis that gave insights into the ultimate origins of these viruses.
All right. Dickson, can you
I will attempt to
read the next one? I know it's
Sure.
very long.
It's all right. I'm a good reader.
All right.
Chris writes, I was excited to hear on TWiV 227 that you and Rich will be in Seattle in June. That's a little... We're late in these readings somehow. And about the potential for a Seattle TWiV episode. Any chance the Seattle episode might be in front of audience? I have been an avid listener since TWiV 145 and would love an opportunity to watch one live. The current weather in Seattle is normally sunny with a high of 20 degrees C, but don't worry, by the time you get here it will be cloudy again with a significant chance of rain. I am a Medical Service Corps officer in the Navy stationed across the Puget Sound from Seattle in Bremerton, Washington. My research interests lie in the convergence zone of virology and immunology. I'm particularly interested in exploiting aspects of viral host interaction to develop new vaccines. I will be applying to graduate school this fall and have spent the last 8 months investigating the virology/immunology research being done in the Seattle area. With that in mind, let me humbly suggest a few potential guests. Dr. Leonidas Stamatatos, is the program director of Seattle Biomedical Research Institute. I know that institute. His work focuses on developing HIV vaccine candidates capable of eliciting broadly neutralizing antibodies, similar to the one used in the vectored immunoprophylaxis study discussed on TWiV 168. His group recently published a study in the Journal of Experimental Medicine in which they demonstrated the removal of specific end-linked glycosylation sites on HIV-1 GP120 allowed the protein to bind to the germline B-cell receptors responsible for the development of broadly neutralizing HIV antibodies. Dr. Julie Overbaugh is a member of the Human Biology Division at the Fred Hutchinson Cancer Research Center, a fabulous picture too, by the way. Her lab focuses on the mechanisms of HIV transmission and recently published a study in the Journal of Acquired Immune Deficiency Syndromes suggesting the plasma level of anti-HIV antibody-dependent cell-mediated virus inhibition in HIV plus women does not correlate to protection from HIV-1 superinfection. Dr. Daniel Stetson is an assistant professor in the immunology department at the University of Washington, where he focuses on innate immune recognition of viral infection. His lab, in collaboration with Harmit Malik, recently published a study in the Journal of Experimental Medicine in which they identified and characterized 5 novel AIM2-like receptors. Thank you for your outstanding informative podcast. It makes the many mile of marathon training all the more bearable. Keep up the good work. Sincerely, Lieutenant Christopher.
Is that what LT is?
I guess.
Yeah.
Cool.
That was a great letter.
Yeah. We didn't do it in front of an audience, but it was Mike Emmerman, and we had Julie Overbaugh and TWiV 143.
I guess he missed it. Although he would have written a letter saying, "Oh, and by the way, I was at your."
He said he's been an avid listener since 145.
Just missed it.
Just missed it. Go back a couple episodes.
Right. Start running backwards.
All right. Let's do one more round of emails. Alan, we're back to you.
Okay. Ken Stedman writes, Ken writes.
That's okay. You're a professor.
We can do
He's a professor. All right.
Because we have.
Not sure if it was a bump, but Jeff Deemer and my paper that we discussed on TWiV 195 was just awarded the BMC Research Award for Best Paper in BMC Journals in 2012.
Wow.
I'd like to think that TWiV had something to do with it. Clear Bump was a very strong potential PhD student who applied to my lab based on TWiV, but he unfortunately ended up going elsewhere. I'll try to say hello in person at TWiV Live in Denver. I'm also using course capture and putting my virology lectures online, inspired by your example.
Cool.
Ken says, P.S. Please tell Dick that we are not kicked out of. We were not kicked out of Edina the last time we were there.
That's great.
I don't get that
It's
either.
a Portland restaurant that specializes in Peruvian cuisine, run by a very good friend of mine, Peter Platt. So
why would he be kicked out?
I have no idea. Maybe they just ended up drinking too much Peruvian liqueur or something. Who knows? But I'm glad you weren't.
This was the paper on he isolates viruses from hot acidic lakes. I think this was Despommier National Park. And we did his paper. They think it's an RNA virus, I believe.
Right.
Is it Lassen National Park in California?
Yeah.
Lassen.
Yes. What did I say?
I don't know. Stassen?
Stassen. He was a presidential candidate, right? Howard Stassen.
Howard Stassen, that's right.
And we did say hello in Denver. All right. Next one is from Manoy, who writes, Dear Vincent, Allen, Rich, Dickson, and occasional other Twivsters.
Oof.
Forgot Kathy.
Yeah.
Yeah.
I would be.
I think his name is pronounced Manoj.
Manoj.
I think.
Okay.
I knew someone else with that name.
Manoj. Yeah, probably. Thanks so much for your wonderfully lively and entertaining podcast, which I enjoy enormously. I work at CDC in Imperial College London, largely doing epidemiology and not lab work, and I find the microbiological aspects of infectious diseases very interesting, though tough to get my head around. Epi is hard enough. I was wondering whether it would be possible to bring on an epidemiologist full-time onto TWiV, because that would open up a wide audience of people who are into virology but find laboratory aspects difficult. With an epi person present, she would ask the questions we would like to ask, and you would ask them epi questions. Your recent conversations on H7N9, novel coronavirus, and pertussis on TWiV were cases in point. They were good, but definitely missed a very important dimension, namely population effects like prior immunity over age, reproduction number, etc. Michael Walsh was great when he was on, by the way, and someone like him would be ideal. I realize you're a virology podcast, but I honestly think you're also filling a vacuum and need for public health broadcasts at the moment. Keep up the great work. Love it when you needle Dickson, Vincent, and when Dickson and Alan debate things like public perception of science. Rich's observations are always sobering, too. I don't have any more computers for another person.
Right. Right.
I can have 6 people would be so many. Right?
Yeah, it'd be a full boat, but an epidemiologist, at least occasionally, we could save up questions or papers, and because it is a nice perspective.
I'm sure Michael Walsh would come on.
Yeah, we could bust Michael back in periodically when we have a particularly epi-intensive topic.
Given my rate of absenteeism, I think you could find room for him.
Yeah, make him the substitute Dickson.
That's right.
And this letter actually points out a couple of topics that we could put on the table for an epidemiologist, to sort of revisit those topics for an epidemiological point of view.
Sure. Mm-hmm. All right, we'll do that. Not a permanent person, but we'll keep Michael Walsh in mind. Kathy.
Adam writes, in TWiV 229, you discuss the development of aerosol transmission in emerging viruses, and specifically the lack of information on how a virus might develop this trait. We've been watching Ebola for the past 37 years, and aside from some rather weak, and to my knowledge poorly reproduced or not reproduced at all, evidence of aerosol transmission in REST-V, so I guess that's the Reston virus. Right. And absolutely nothing to my knowledge in EBOV, SUDV, BDBV, which is Bundibugyo, or TAFV, Thai forest virus. Of course, we see the same problem in H5N1 outbreaks. Do you think that the relatively low number of passages is preventing the required mutations from appearing in the same virion at the same time? Or is this a fundamental biochemical issue? Furthermore, if low passage numbers are the issue, and if H5N1, which has a genome in the neighborhood of 100 times less stable than that of any Ebola virus, and so if H5N1 has not been able to achieve the magical combination of mutations required for aerosol transmission, what chance does an Ebola virus have? Of course, one would assume that the mutations required would be different.
I think it's a problem that the virus cannot sustain the right changes to transmit by aerosol.
I think it's a fundamental biochemical issue. We don't really understand what mutations are required, and they're likely to be different for these different viruses. I think it's a complex biochemical issue. That's why the transmission experiments are important.
I mean, the ferret experiments show that you can select for an aerosol-transmitted H5N1 among ferrets, but it loses pathogenicity. It has a fitness cost. So in nature, that virus probably doesn't get it.
We're never going to see it. Yeah. Right.
Yeah, I think it's an essential, a fundamental trait of that particular virus in its current state. Now, whether it could get beyond that, I don't know. People tried with ferret experiments and got to the point where it does impose a fitness cost. If it were easy, it would have happened already.
I think so. We could be wrong, though.
We could, of course, yeah.
Rich, you're next.
Stephen writes, while not virology related, this item may be of interest to the TWiV crew. Kathy picked the Finkbeiner test in TWiV 223 and, of course, science writing as an alternative career for scientists and as part of public education in science comes up sometimes. So this is an article to the closing, a link to an article that discusses the fact that a very good graduate science writing course in Johns Hopkins is shutting down. Okay, due to apparently lack of enrollment.
Gee whiz.
Yeah. Do you know anything about this, Alan?
Yeah, this when this happened, it was obviously quite the talk of the science writing community because an awful lot of people graduated from this program. This is one of the big science masters in journalism programs. There's Hopkins, there's NYU, there's UC Santa Cruz, I think it is, and a couple of others. MIT has a good one. There aren't a lot of these sorts of programs around, and this one's been around for a while and has produced some incredibly good writers. And they shut it because they weren't getting the number of applicants they wanted to see in order to make the program sufficiently selective. At least that's the cover story. I don't know if that's the entire reason. I'm sure there's at least a little bit of academic politics behind it as well. But things in journalism, if I were just coming out of college now and looking at careers, I don't think I'd find journalism very attractive.
Right.
And I don't think I would find going into more student loan debt for an advanced journalism degree to be a very attractive option. And the folks who had other careers, PhD, and then went to science journalism schools, some of them are deciding on other tracks as well. It is a tough line of work to be in right now, and I think that's probably what's affected enrollment figures.
And yet there's no greater need for accurate science reporting than there is today because of all the confusion out there.
Yeah, that's the irony, isn't it?
Yeah, it is.
You'd think there'd be demand. There is demand.
But look, ABC is a good example of that then. Who do they turn to for their information? Come on.
Right. There is actually a huge population of people who are interested in science and who will seek it out. And they'll seek it out online, and there are science blogs that are doing great, and there are lots of writers out there who are writing for that audience. But the step that hasn't really quite been made yet is how do you make it pay in the online world?
Right.
And the nature of the web is that everybody wants everything for free. Nobody likes to look at ads. And so, well, that's lovely. I don't like to look at ads either, and I want everything for free too. But if you want professional quality content, somebody needs to pay a professional to produce it.
You just said the magic word, though, Alan, because what the papers are looking for, at least in the common popular press, is sensationalism. And science writing is not sensationalistic, no matter what.
Well, that may be true in the business office, but in the editorial office, I do believe that there's a real ethic of wanting to inform the public, whether the topic is politics or the environment or science or anything that's going on. The people who end up in journalism really doing it and editing and writing and producing video and that sort of thing, they do it because they want to tell people stuff.
No, I agree. So name another newspaper besides the New York Times that has a Tuesday relevant section called Science Times, Science News, Science,
Right. No, that was always.
Is there another newspaper that has that though? I don't know of any.
I don't think there's one that's as developed, and the one in the Times has shrunk and become essentially the Times medicine section.
Something like that. That's right.
There is a real problem making science journalism pay.
Right.
At least at the levels that something like a newspaper needs in order to keep it going, especially when newspapers are in the process of collapsing entirely.
Sure.
And then the standard company line is, well, that's all moving online, so the future is online, but nobody's figured out how to make online journalism pay.
Right.
Not in the scale that it needs to. I mean, individual journalists are still making money, and I still get checks that clear.
Maybe we need Mark Zuckerberg to develop a new model for it.
I would not trust him to pick up my dry cleaning.
Oh, okay.
You saw what the Chicago Sun-Times did recently. They fired all their photographers and gave iPhones to their staff writers.
You're kidding.
They said, take pictures.
Oh, my God.
Yeah, because photographers are too expensive.
Oh, my God.
The last one, Dickson, you have the honor of reading.
Okay. So, Luis writes, Dear TWiVers, thank you very much again for your wonderful podcast. I haven't missed a single episode since December 2010 when I was introduced to TWiV. In TWiV 230, I was happy to hear that Vincent had chosen The Origins of AIDS as a pick of the week, since this was my pick back in TWiV 191. Certainly, it is well written and an entertaining book, and very interesting from a scientific point of view. Vince's choice was a relief for me, since after my recommendation I read some critique which was not as positive as mine, and he gives a reference to that. Today I have just read a short commentary about additional experiments following the high-risk flu saga. Single gene swap helps bird flu virus switch hosts. Another reference to it online. I haven't got access to the paper yet, probably due to embargo, but I am sure it will provide an interesting discussion for a coming episode. If I understood correctly, one of the issues not fully understood refers to the relevance of the animal model, since in this paper guinea pigs are used instead of ferrets. Finally, I want to suggest another pick of the week. Is that appropriate to continue on? This time, it is the Public Health Image Library, in parens Phil, of the CDC. It has a nice collection of illustrations and videos and can be an interesting source for educational purposes. I am sorry if it has already been chosen, but in any case, quoting Andre Gide, everything has been said before, but since nobody listens, we have to keep going back and beginning all over again. Is that why you had me read that? Again, thank you very much to all of you, Kathy, Alan, Rich, and Vince, and the guys who's reading this right now, for your great contribution to knowledge dissemination and for stimulating the young generation of future virologists. Best, Luis. P.S. Today we have a partly cloudy day, but sunny day in Madrid with an actual temperature of 18 degrees C, 64 F at 5 p.m. local time. Thank you for that addition. Well,
this paper he's talking about, we did on a previous TWiV.
We did.
I don't remember which one. But
so we call ourselves the Tandem Repeats, that's all.
No, it's okay to repeat things. It's not a problem, but I guess he wrote this before we did it.
Right.
What would that have been? Does anyone remember? Yes, TWiV 229, partly cloudy with a high of H7N9. That's a great title.
Thank you, Alan.
Put that link in.
And then I think maybe Rich picked that Public Health Image Library once before too. So
before he started listening.
Right.
It's okay.
But he's absolutely correct. It's worth picking again because it's a great site.
Yes.
And everything has been said before, but since nobody listens, we have to keep going back and beginning all over again.
I love it.
I mean, I didn't even remember, right? I had to go look it up.
Yeah, I forget that site all the time, and then I'll be scrounging around for a picture somewhere, and I need something that's got public domain rights or Creative Commons rights on it, and then I remember, oh, right, the CDC, it's all public domain, and they've got these great slides online.
That's right.
All right, time for some picks of the week, Dickson.
Uh-oh.
Do you have a pick?
No, I don't. I don't.
Didn't you come up with one while we're doing
I can
this?
pick one I've already picked, though, because apparently we're supposed to do this.
You're such a cheater.
No, wait. I have, I do a lot of surfing too, guys and girls. Whatever.
I didn't picture you as a surfer.
No, I surf. And
Dude.
yeah, and I hang 10 sometimes too. So one of the sites that I've picked again and again and again, because I'm interested in a larger concept called diversity of life, obviously. And I used the book by E.O. Wilson as part of my teaching exercise to my students when I was still active as a professor. And one of the sites that I just loved going to again and again and again is if you go to Google Images and type out mantids, MANTIDS, just do that. And you can revel in the diversity of life from the perspective of praying mantises, or manti, however you wish to do that. I've got a collection of high definition images taken by fanciers of mantids that I just am absolutely stunned by, particularly the orchid mantises.
These are beautiful.
The orchid.
Yeah.
So it turns out that Dorothy McClintock had on her desk
Barbara
and her daughter Barbara. 2, 3, 4. I remember, I haven't been on this show for a while. Give me a break here. She had one of those orchid mantids in a terrarium, and she jokingly said that everything she believes about evolution is true except for this organism. She thinks God created it because it's just so incredibly beautifully different than anything else she's ever seen. And the mantid sits inside the orchid and pretends it's part of it, waiting for its prey to come along. I think that's fantastic.
Ooh, there's a couple of really big ones here.
Yes, there are. Yes, there are. So
that is scary.
Having just got back from Singapore, if I were to have gone north into the Malaysian jungle, I would. That's where they apparently have the most diverse numbers of mantids. So that's my pick of the week.
There's a picture of somebody's hand with a couple of these.
No, some are enormous, but they're friendly to people. They don't eat too many people. Actually,
Ever been bitten by one?
No.
Oh, they bite.
Do they?
But only when you're trying to shove them into a killing jar.
Oh, that's.
Did you see any bugs in Singapore, Dickson?
I didn't see any bugs, but I visited the orchid herbarium at the botanical garden, and I was just blown away by the number of species that are able to grow in that atmosphere. They just leave the doors wide open, and they just grow like wildflower, and it's just unbelievable. The ginger plants and the heliconia type flower plants that I saw there were just amazing.
The orchid mantises also grow like wildflowers.
Thank you.
I went to I googled orchid mantis, and there's some gorgeous images there too.
Yeah. It's a fantastic species for teaching biodiversity to a mixed audience, like little kids, for instance. You think there's only one species of people, and that's true. Look at how many different species of mantids there are.
Yeah.
And then it allows you to go back and ask the question, how come there's only one species of human? And then no one comes up with the answer, of course, including myself.
Thank you, Dickson.
You're
See,
welcome.
it wasn't so hard, was it?
Yeah. No, not at all.
Rich, what do you have for us?
I've had this on my list for a long time. Probably a lot of people out there know about this, but I subscribe to, I guess, a Facebook page called Science is Awesome.
That sounds great.
And I don't think you have to be on Facebook to access this. I'm not sure.
I'm looking at it right now, and I am definitely not on Facebook.
Okay. So if you use this link, you're okay. Otherwise, you have to be a little careful, because if you Google search Science is Awesome, you come up with a couple of different Facebook sites. So you got to make sure you get the right one. This link actually is Science Seriously Awesome, blah, blah, blah.
Science is seriously awesome on Facebook, yeah.
At any rate, this started under a different and profane name that the followers managed to persuade the author to change.
Oh, this is that one.
Same one. And I just like it. You know, it's got lots of different content of really cool little bits from science, and they come up for me all the time, and I think it's really good. It's founded and moderated by a woman named Elise Andrew, who's employed by LabX Media Group, which owns Lab Wrench and publishes Lab Manager Magazine and The Scientist. And she does a great job with this, I think. So just check it out.
Wow.
Yeah, I put a link to TWiV a couple days ago on it, because you can put links. It's not limited to the admins.
Good.
But it's already disappeared since there's so much activity here. Oh, well. Kathy, what do you have?
I picked a PhD comic, which I think from its date is a little bit old, from 2011. But someone sent this to me, and the gist of it is it's a flow chart about what to do if you haven't received any email in the last hour. And step one is you panic, and then you can send yourself an email just to make sure that things are working. And then, but maybe it only works for you. And so it's really kind of funny, because I think all of us have had that experience of nothing's happened. Maybe my software is not working. Maybe my server is not working. And I just thought it was really well done.
That's good.
What I liked about this is that all of the options that say that everything is actually really okay are non-viable options. Right? The assumption here is that something's really wrong.
Yeah.
Right.
Alan, what do you have?
I have an entirely self-promotional pick. I decided to split my science blog onto a new site. So the alandove.com site is my personal site, and I'd been doing my science blogging there, but my relatives want to see more pictures of my kid, and the people who want to read my science writing probably don't. And so I bought a new domain and set up my science blog, and I'll be doing science posts there. It is called the Turbid Plaque.
All right.
Nice.
Cool. Very cool.
All right, so that's the end of alandove.com, right?
Well, alandove.com will now be it'll echo posts from the Turbid Plaque, but it'll also have a lot of stuff that is probably not of interest to TWiV listeners.
The Turbid Plaque is a blog about science, the people who do it, the stories we tell about it, and how we react to it. It won't be exhaustive or authoritative, and it will get messy sometimes. Okay.
I love your subtitle here. Life is complicated. Very good.
Who have I left out? Kathy. We got Kathy.
You.
Me.
You.
My pick is a publication of the American Academy for Microbiology. This is a part of the ASM, which does a variety of things, including running what are called FAQs, frequently answered questions. They have colloquia where people get together and talk about a topic, and then they publish the results as an FAQ. And their latest has just come out. It's on West Nile virus.
Right.
And they are free reports that you can download.
Cool.
And very nicely done.
Beauty.
With experts.
Yep.
And it
Nice.
tells you all about West Nile virus,
Yeah.
what it is, how we got it here, and what it does, blah,
Nice.
blah, blah, etc.
Nice.
Really nice illustrations, which are useful for teaching, like that one there.
Right.
Chance of a mosquito picking up West Nile virus from an infected host,
I
depending
see
on
it.
how much virus you have in you.
Right.
Like that, Dickson?
I love it.
Yeah.
Speaking of that, a friend of mine and I, who she also listens to TWiV, we like to go back periodically and listen to TWiV number 1 because it's just great. It's
really? I thought it was terrible.
Oh, shucks.
no,
Shucks.
it's Vincent and Dickson talking about West Nile virus.
Yeah.
Right.
After that, when Dickson said, "That's it, I have
It was
nothing
all
more to
downhill.
say."
That was my only touch
I
point.
get I cringe when I listen to number 1. Rich, you and I listened to it in the car when we were driving to
That's right.
Funny.
Florida Gulf Coast, right?
Uh-huh.
Maybe we should go back and do another TWiV number 1.
No, you can't. It's done.
Sure you
but,
can.
It's
No,
just,
we
the
can do-
sound is not very good. Remember, we had technical issues. It was
That's
in
right.
your
And
crappy office.
We had not yet become abusive.
Yeah, we were actually friendly back
That's
then.
right.
What happened?
I don't know, but
Familiarity.
does.
breeds. You know what they say, familiarity breeds, but we
Yes.
won't go there either.
Oh.
This is really quite a publication that the Academy puts out.
It's very nice. This is from a meeting where these people got together and talked about things, and they put it all together in this
Nice.
colloquia. It's good.
Was the meeting specifically aimed at producing this document?
They are all aimed at producing a document. So they get
Okay.
people together and talk about things. They don't give talks. They just sit around a table and talk about the issues surrounding here, West Nile. Someone takes notes, and then the staff makes figures. It's a lot of work.
Wow.
But all the FAQs that they run, and they're on all areas of microbiology, end up making a publication. So you can go, there's a link on the page,
I'll be darned.
browse all reports.
Yeah.
They've got them all, Bacillus thuringiensis, the safety of water, genome annotation, antibiotic resistance, etc. There's lots
You
of
look
them.
at the participants here; they've got everybody who knows anything about this, so it's it's a really authoritative,
It's very good.
and it's beautifully laid out. It's really nice.
Yep. These are little known things that are very useful.
Not anymore.
So-
Everybody knows now.
Right.
Everyone should know. Everybody in the world now
right.
will know. All the billions of TWiV listeners.
Yeah,
Maybe we should send one to Jenny McCarthy.
She doesn't read. What good would it do?
Sorry.
Maybe she doesn't know how to read.
They don't need to insult Dickson, but you do need to be.
We're not
incisive.
insulting. We're not insulting.
Well, you said
here.
she couldn't read. I'm sure she can read.
You think?
Yeah.
She just chooses not to
Just
pay attention
let-
to certain things.
All right. We had a listener pick from Luis, I think. Is that his name? Yes. Luis from Spain had a pick, which we will use.
Right.
And that brings us to the end of number 242, which you could find at twiv.tv and also on iTunes. And as always, if you like us, go over to iTunes and subscribe and leave a comment or a rating so that more people can find us on the front page of that podcast directory there. And send your comments and questions to TWiV at twiv.tv. Right, Dickson?
Right.
Where can we find you, Dickson?
Usually not here.
Yeah, he's moving all the time.
No, I'm at Racaniello.org.
Yes.
I'm at medicalecology.org.
I thought it was .com.
No, it's org. And
Okay.
it's verticalfarm.com.
How is the vertical farm?
You know, it's fantastic. There are lots of them up now and running. It's too many to keep track of now, and it's been quite an amazing
And
journey.
so you were wealthy as a consequence.
Nothing, not a penny. Nothing. I have no skill at that level. I do love talking about it, though, and I love visiting them. And I when I was in Singapore, I met the guy who runs the one in Singapore. His name is Jack Ng, and he started out with a 2,000 square foot vertical farm 2 years ago, and it became so successful. Now, remember, Singapore is a space-challenged area. It's not very large. He now has increased his production by tenfold by increasing his footprint by threefold.
Wow.
It's been fantastic, just absolutely.
Great.
And they love it, and he sells all he can grow.
Great.
Yeah, so it's good.
Thanks for joining us today, Dickson.
It's my pleasure.
Always good to have you.
My pleasure.
And you're around the rest of the summer, aren't you?
I am. I'm here.
So join us again.
Of course.
I'll get another computer so you don't have to sit next to your huge iMac.
I
there.
love sitting next to you, Vince.
Kathy Spindler is at the University of Michigan. Thank you for joining us, Kathy.
Thanks. This is fun. I learned a lot.
And we will see you in person soon,
Yes.
as we
Soon.
will see Rich Condit at the University of Florida, Gainesville. Thank you, Rich.
Sure enough. Great time.
You're going on Saturday as well?
I am leaving on Saturday. I will be there in time for the keynote address Saturday night.
All right. See you there. Alan Dove is at turbidplaque.com. He's also on Twitter. Thank you, Alan.
Always a pleasure.
I got Turbid Plaque now in the show notes.
Yes, I see.
So we will get lots and lots of visitors to your site. So start writing.
Yes, I'm gonna be blogging there.
Very good. I am Vincent Racaniello, and you can find me at my blog, which is virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. You like I Want My MMTV?
I do.
I don't even know what that means.
Don't you know who Dire Straits is?
Yeah.
It's a group.
What does it have to do with
Back when
MMTV?
MTV first started,
I want my MTV.
Yeah.
Come on, we can do harmony here. Come on. We're a group.
I think it's a limited thing.
No.
You guys like it?
It would appeal to a younger generation than some of us.
You guys may remember a long time ago, Adam. What was the first MTV jockey? What was his name? Adam.
Oh, didn't even
Yeah.
Oh
watch it.
yeah,
Never watched
yeah. We had some kind of a dust up with him, didn't we?
Yeah, he has a podcast called. Oh, come on, Adam. Not Corolla. No, Adam and John. What's the guy's name?
Curry. Adam Curry.
Adam Curry. Adam
Google
Curry.
to the rescue.
He has a podcast called No Agenda. And someone sent us a clip of No Agenda where they were criticizing some vaccine. So we criticized them, and
I
Adam
remember
Curry.
Oh, yeah, we got into a pissing
So actually, we
contest.
got a lot of listeners from that.
We did.
So he was one of the first video jockeys on MTV.
Yeah.
And this is really a Dire Straits reference.
It is.
Yes.